Today I'm going to talk about the dynamics of influenza A infection in wean-to-finish populations. This is how my presentation is going to be divided. I'm first going to give a brief introduction on influenza viruses and swine population, then present my objectives, materials and methods, results, the working process that I'm doing right now, and finally the conclusions that we have from these results. So we know that influenza is an enveloped virus, single-stranded negative sense RNA virus, and very importantly, its genome is segmented in 8 different genes that translate up to 12 different proteins. From these proteins, there are 2 main antigenic proteins in the surface of the virus, the hemagglutinin and the neuraminidase, and we know that there are 17 and 9 different subtypes for each of them, which allow us to classify different viruses in different species. The overall diversity of influenza viruses is defined or is driven by 2 different processes, antigenic drift and antigenic shift. Basically, in antigenic drift, what happens is that mutations accumulate over time within a gene, and that makes virus different. So what I want to illustrate here in this phylogenetic tree published by Nelson and co-authors is that different viruses can evolve over time, and basically they can also be associated to swine movement between different regions in the United States. This is the case for H1 viruses that have been evolving from 2004 to 2009 and have been moving from the southeast region to the Midwest, as well as from the south central to the Midwest. The other mechanism by which influenza changes over time is antigenic shift. So remember that I told you that the genome of influenza is segmented. So when host is infected with 2 or more different viruses, it can exchange different gene segments from with different origins. So basically, what I want to illustrate here is that swine viruses have evolved over time, and after many or few spillovers from different species, they have evolved to what we have currently circulating in swine population. So it's important to take into account that these 2 phenomena can happen at the same time. While the virus is changing with antigenic drift, it can also be swapping genes with other viruses and increase the diversity of swine influenza. So we know that the virus is worldwide distributed. Basically, I would say that it will be pretty easy to find influenza in swine populations, but I am not sure if we really know if the virus follows endemic or epidemic pattern within swine populations. We know that it's a primary cause of disease. It is also associated to the respiratory disease complex, and it can reduce feed conversion and average daily gain, and increase the number of days to market. It is an infection that has a high morbidity, which means that once a virus gets into a population, most of the animals are going to get infected, but it has a very low mortality. Influenza viruses, the swine influenza viruses, are also a public health threat. Unfortunately, the pig has been blamed to be able to generate human influenza viruses with pandemic potential, and we need to follow up and assess the risk of really of this situation to happen. So a little bit more on influenza A diversity. This is part of the study that Matt mentioned in the previous talk, where Caesar followed different growing farms, growing to finishing farms, and they were able to find, each line is a farm and each dot is a sampling. Black dots indicates that they were negative, but what I want to show you here is that there's some farms that where they were able to find influenza in several different points in time, including different subtypes, like different colors, but more interestingly, some sampling periods where they were able to find different subtypes of influenza at the same point, which means that if antigenic drift is happening mixed with antigenic shift, well, the diversity of, or the likelihood of influenza changing over time is extremely high. To mention a little bit on influenza transmission in pigs, we know that the main route of transmission is direct contact, but airborne transmission and through fomites have also been reported. And we know that the transmission rate estimated by the R0s, which indicates the number of animals that can be infected after a single introduction of a positive animal into a population, can have different values depending on the immune status. So we know that in naive population, this R0 is pretty high, meaning that once an infected pig gets into a naive population, the infection is going to spread really fast. But with different immune status, that R0 is going to change. So if the R0 is greater than 1, then the infection is going to spread out. If it's lower than 1, the infection is going to die out. So in both studies, in these studies with active and passive immunity, they found that when pigs had an homologous immunity to the virus, the infection will die out with R0 lower than 1. But with different, with an heterologous vaccine with different immune status, the R0 can differ depending on how close the vaccine virus is to the challenge virus. So with that information in mind, our research questions are, how is influenza virus maintained in swine population, and what is the role of weaned pigs in this process? What is the transmission and infection pattern of influenza in weaned populations, and how does influenza change over time in endemically infected populations? Therefore, our objectives are to characterize the evolution of influenza virus infection and transmission in weaned pigs, to explore the transmission pattern of the virus in weaned pigs, and to understand the association between the virus exposure, immunity, and infection after weaning. For that, we designed a cohort study, prospective over time. We selected one endemically infected wean-to-finish farm, and by endemically I mean a farm that we knew that tested positive on a regular basis. Located in Minnesota, it has 8 barns, and it housed around 2,000 pigs per barn. This wean-to-finish unit has a single source of pigs, and we also knew that that source was positive for influenza. So we randomly selected 132 pigs at weaning, at arrival, expecting to find at least 1 positive when the prevalence was higher than 2.5. We identify an ear tag each pig. We went to the farm on a weekly basis for 15 weeks, so this is at arrival. We collect nasal swabs and oral fluids on a weekly basis, and we collected serum samples at arrival, week 4, 8, and 12 after weaning. We tested all the nasal swabs and oral fluids by RT-PCR, considering them positive, suspect, or negative, depending on their CT value. We test all the serum samples by ELISA, and again consider them positive, suspect, or negative based on the sample to negative ratio. We estimate the seroprevalence in the different sampling point times of our study, and we compare the mean change of this SN value over time. And we also estimate the effect of being seropositive at weaning on the seroprevalence after weaning of those pigs. We estimate the weekly prevalence and weekly incidence. The weekly prevalence defined as the number of positive cases by week. And we compare those numbers to see if we have a higher or lower number of expected cases by week. Since we knew how many pigs were positive and negative each week. So we were able to estimate the incidence, which is going to be defined as new cases per week. And we also studied or looked at the possibility of reinfections, meaning that a pig tested positive in 2 or more weeks, in 2 or more non-consecutive weeks. So what do I have for this study? So when we looked at the ELISA sample to negative ratio, here I have the value for the test, and here are my 4 different sampling points. So first, I want to illustrate that there is a higher diversity of the antibody levels of pigs at weaning. More than 50%, the black line indicates the median, more than 50% test positive. This is the cutoff value for positive, below 0.6 is positive, and between the 2 lines is the suspect region. So as expected, most of them lost the maternal antibody and became suspects 4 weeks after weaning, and most of them have a very strong response to influenza 8 and 12 weeks post weaning. And when comparing the medians between the different samplings, we found out that they were all different, statistically different. However, what I want to show you is a little bit more on detail what happened with the ELISA results. So this is each box represents an animal. I have 5 animals that died, so I didn't include them in these results. So each box represents an animal. Each dot represents the result for each ELISA. The red lines represent the cutoff value between negative and positive, the positive being underneath the line. And in the x-axis, I have the 4 different sampling times. So here, these are the animals that started being positive at weaning, and you can see that most of them have sharply increases in the SN value, indicating that they are losing the antibodies. However, I want to illustrate here in the bottom, there are a few animals that they didn't lose, or they didn't lose the maternal immunity, indicating that they got infected in the sow herd and developed active immunity, and they are keeping it, like this one here, or this one here, are keeping that active immunity through the wean to finishing period. Up here, I have the 6 animals that tested negative at weaning and how they gain active immunity pretty fast, and everybody else, like the remaining pigs, are the remaining boxes. If we summarize that into 3 different plots, we can see those that started positive at weaning, suspect at weaning, and negative at weaning. And when we compare the SN values at 4 weeks between groups, I find out that when they were positive at weaning compared to the ones that were suspect at weaning, these 2 means were different, but they were not different compared to the negative. But these negatives are acquired immunity, and these suspects, are acquired immunity, and these suspects are still probably maternal immunity. These 2 means were also different, but all the other comparisons that I did between groups were not different, indicating that at the end, all animals were infected and probably got active immunity to the infection. From the PCR results, I have 127 total included for this analysis. In red, I have those that tested positive by week, and in blue, I have those that tested suspect. When we compare the number of positives, the frequency distribution by week, we find out that they're higher at week 2 and 7, indicating that we truly had 2 epidemic waves during the wean to finishing period. And if we are a little bit conservative and just take into account the positive results, we found that 99 pigs from the 127 study tested positive at least once. But very interestingly, we found that 29% of those pigs tested 2 or more times positive in non-consecutive weeks, indicating that reinfection was happening. But since we consider that the suspects, the blue ones here, are true positives, there might just be in the tail of the infection and be shedding lower amount of virus. We find that 124 pigs, 98% got infected, tested positive at least once during our study, and 82% of them retested positive in non-consecutive week. And this result makes more sense with our ELISA results, where most of our animals had antibodies to influenza. Now, when we look at the prevalence and incidence rate for our 15 weeks, we find that the prevalence ranged between 2 and 64, but with a high value also in our second epidemic wave. We found that our incidence rate by week was not constant, that it ranged between 1% and 52%, with a high value again at our second wave of our second epidemic wave. But for most of the weeks, the incidence rate was below 6%. Now, when we look at the incidence, the cumulative incidence rate, we found that most of the animals have tested positive after 2 or 3 weeks, and after that, we start seeing how we got reinfections or animals that tested more than once in non-consecutive weeks. In the last column, I have something that is still work in progress. Some R0 estimates for the values for the periods that we have here. So we know that 19% of the animals are coming positive to our population. That's 25 animals. And after 1 week of commingling with the other ones, 50% of my animals became infected. We can estimate that 1.6 animals got infected for each animal that was being introduced positive in the population. Then when estimating the same R0 and kind of see how the epidemic is moving or how many different animals are getting infected across time, that R0 go down to 1.2. If you remember the R0 that I mentioned for naive population, it was around 10, indicating that the maternal immunity did not prevent infection, because indeed the animals got infected, but it actually reduced the rate of transmission among these pigs. So what I'm doing right now, we selected 96 samples from the positive pigs, especially those that tested more than once, for whole genome sequencing, whole genome amplification and sequencing. And what we want to do is assess the antigenic drift and antigenic shift to see if the virus is changing over time, and those little changes or huge changes, I don't know what we're going to see yet. But if those changes are allowing the virus to maintain in the population, or if there is a new virus that is coming in, remember that this is a wean-to-finish unit, and there are more pigs around, and eventually a new virus could have jumped into the population. We are going to select, depending on the sequences and the data that we find here, viruses as a reference virus for each week. And we want to estimate the real exposure by HI reactivity of the serum samples to these reference viruses, and eventually also explore the antigenic differences of the reference viruses across time with antigenic cartography. In conclusion, I can tell you that wean pigs can be a source of influenza to other swine populations. That's kind of a proof of concept. We know that pigs can bring viruses from the sow herd to the weaning and later stages. The presence of maternal immunity in weaned pigs is highly variable, and this maternal immunity did not protect weaned pigs from being infected, but it did reduce transmission. Maternal immunity at at weaning was correlated with antibody levels at 4 weeks, but not at 8 and 12 weeks post-weaning. The interaction between influenza virus transmission, infection, and maternal immunity allowed for 2 epidemic waves of the virus in less than 8 weeks, which I think is very interesting. And our results indicate that pigs can be reinfected over a short period of time, and as a result of that, influenza virus can remain in the population. At the end of this study, all pigs tested positive to influenza by ELISA, indicating that all pigs were infected at some point with the virus. With that, I would like to acknowledge my advisor, Monsi, and all the other professors that have collaborated with this study, Pipestone Veterinary Clinic, and especially Dr. Spencer Wayne, who helped us finding the farm and doing the study.